Okay. Good morning, everyone. Today is great Friday, like time for starting of the summer. I have a tropical speaker, a dinner speaker, but he's based in Bristol. But we have this rule that we create to help each other that if the sunlight is above 25 degrees, at least in the day, you have to start this before. You get ice cream if you arrive before the sunlight starts. Okay. Oh, and those the last one. I can get another one for you. Yeah. But so we have today's Hermes Bloomfield-Gadêlha. He leads the polymath lab at University of Bristol, but before that, he was also lecturer at University of New York, and before that, he was research fellow at Oxford University, and before, let's see if I forgot that, Cambridge, and a PhD in Oxford, and before that, that's when we met. He was undergrad in Brazil, in Recife, where I'm from, so that's some friends. There's also some work together in Fox Space that also work.
Nice.
So it's a great pleasure to bring Hermes to speak. But he speaks in his broad field of self-organization scene there in Florida. Hermes, thanks for coming.
Wow.
The floor is yours.
So thank you, Fernando, for this opportunity. He has been trying to get me to give a talk here, I think, for the last 5 years. We had the pandemic, and then I had a second pandemic, like 2 children. And basically, this is going to be my second talk in a department after 5 years.
Whoa.
Yeah. So I forgot what we do. So sometimes I talk too much. So feel free to just but also I'm very excited because it's so rare that I have this opportunity, like this is kind of, oh my God, it's amazing. Yeah. And going back, so Fernando and I, we met when we were undergraduate students. So when we were 18 years old, and I never thought, at that time, that time ticking will be a completely different place in this planet, but being able also to share the research we do, that was really nice. I think there is a very nice symmetry there that is quite cool. So I'll introduce a little bit what I do in Bristol. So I'm housed in a school that's called Engineering, Mathematics and Technology, and my lab is called Polymaths Lab. The reason why is because we receive students and fellows from all different backgrounds. So I have students that will be working, for example, in more experimental aspects, others there will be more theoretical aspects, others will be working on data aspects. And of course, the combination of all of that. And then when it comes to experimental as well, there is a lot of variation. I have students that are medical doctors, for example, working in medical robotics, but also have people in aerospace engineer, physicists, traditional physicists, mathematicians. So it's really difficult to define what we do in the lab because we kind of work with many branches. But what the most important thing is that we meet the same focal point. And the focal point for me is the mathematics. So I'm going to explain a little bit of the structure. So I already said we house in this wonderful place. So Bristol, if you're interested, contact me. It's really a wonderful place for Nice. Cool. So I'll put in the presentation mode.
Sorry about that. We lost connection.
Sorry, we're back. Oh, I can see the camera is also here. Maybe I should see now. Okay. Yeah. So I strategically moved to Bristol because there is all this momentum in robotics and as a mathematician, I wanted to take advantage of the robotics, especially the soft robotics. So my lab is housed in the soft lab in Bristol, part of the Bristol Robotics Laboratory, but also we have a lab spaces in the medical school because I work a lot with biological and medical problems. When it comes to the actual resources that we have in the lab, it encompasses things like high resolution, high speed microscopy. So I'm very interested on the very small, and that happens very fast, which brings a lot of challenges in like a double, because you have time and space. Then microfabrication and manufacturing, 3D printing, bioprinting. So these are more experimental techniques that we use in the labs. And of course, going towards more micro-robotics, biorobotics, software creators, and how we... so these are basically the kind of things we use at the experimental side. And the mathematics is the central bone, is the backbone of all this. Because as a mathematician, I use maths to make things, but make things through the experimental setup. Measure things. So if you do experiment, you want to squeeze your data and get some numbers. Understand and predict. So either you use model to predict or to understand, or both. And I'll show you some examples of how we use this in a holistic fashion. These are some of the things that we do. I will not be talking today, okay? But I'm presenting here more as a potential collaboration. Could be that you see a particular toy, a particular system that interests you that I don't know of, but I'll not be talking about today. So 3D printed microscopy, we do a lot of 3D printed imaging. So make our own microscope that is cheap, that I can send to anyone. Microtraps, acoustic manipulation, so you use sound to move objects. I already mentioned bioinspired and microrobotics. Cilia, I'll be talking about a lot today about cilia, acoustic flows, and more large-scale robotics, fluid structure interaction problems, and computer vision, so image data analysis in this kind of form. So some of these examples, for example, I have students where they're working on AI to capture better how microorganisms move. So this is a good example of a sperm cell moving swimming in a fluid. But if you segment, you can lose track of the cell. That's really hard. So using segmentation-based approaches is just almost impossible to track these things. So we're taking a completely different perspective and saying like, okay, can we use some forms of machine learning to actually not have to deal with this horrible segmentation problem. As I mentioned before, when I started my PhD, I did a lot of experiments myself, but I was very frustrated to always be hostage of people that have their own labs. I always wanted my own microscope because I wanted to try different things from the agenda that, most of the biophysicists, I just wanted to try different things. But who on earth would give a mathematician a microscope? No one. They're very expensive. Nowadays, they're cheaper. So I said, you know what? I want to make this cheap. So this costs 30 pounds. And because it's so small, you can put anywhere. You can put in an incubator. But you can do crazy things. The things I want to do is basically take these. I have a couple of students working on this. And you throw out of the window. so that you see microscale in a free-falling condition. Something that would be impossible to do with a normal microscope, or acceleration, or microgravity, for example. So now we're playing with these in different ways, as I said, as a crazy person would do. But you would not throw like a 70K microscope out of the window, but this one you can. And, and then you can put in drones, you can put like, so we have projects where my students are trying to put this in a little boat so that we have like real-time water surveillance in rivers and ponds and things like that. So to increase the amount of data that you generate. Microfabrication for me as well was a big bottleneck because it's too expensive. Lithography, clean rooms, I don't know how familiar you are, but it's very expensive and you need also specialist people. So we came with a methodology that we can print microfluidic chips, straight from the printer, straight from the 3D printer. So you don't need to go to lithography anymore. So you can take your mold, and this is a good example of making microfluidics with something that we call agarose. Agarose is a biocompatible hydrogel that allows you to do biological assays that are more fitting with that basically doesn't interact as much badly with cells and so it's more biocompatible, for example. And that is possible because we 3D print first and we create a stamp, and then you stamp the hydrogel, and then you're good to go. So this traditionally will be so expensive. If you go to any company, they'll charge you at least 5K if there are many ifs as well, and so forth. So now we are in the point where we have this toy, and now we're moving towards, okay, what are the physical systems that we can exploit this? And this is a good example where we created these arenas where sperm cells, they can move, they can come here, they get trapped, and now I can change the conditions and keep imaging the same cell, what happens. So the kind of things I'm interested are physical interactions. For example, what happens if you increase viscosity, change temperature, put a chemical, reduce this chemical, but you see the changes at the individual level. You see the changes at the individual level, not at the population level. So that's quite cool. And this is the other project I mentioned about acoustic cilia. So basically, we use very cheap transducers that vibrate very, very highly. And then if you make these structures, because they are almost rigid structures, they will vibrate as well. They vibrate so fast you can't see with this camera. And it emulates the flow fields of cilium, which is what I'll be talking today as well. But the good thing is that you tune on and off these, you have a huge amount of control. You can see that you have a flow that is coming in this direction. So we can now use these to control the microscale as a computer as well, or even to have experiments in which the experiment is what I call is a live experiment. As you measure, the measurement changes how you interact with the experiment, and this becomes an emergent. You produce a feedback in the system. So you make more intelligent systems with this kind of thing. And these are all the examples where if you use bubbles, then the vibration becomes way stronger, and then you can create large scale flows. So here in just tracer particles, here I have a bunch of sperm cells that are rotating like a washing machine, and we can rotate so fast that we can heal them. Which is quite interesting because then we can test what is the physical limits that these microorganisms can withstand, and among other things. So you can see here is a bubble, and you have these other micro beads that are bouncing, and as soon as we ramp up the frequency, you completely lose the motion. But here we are in the order of gigahertz here. and segregation of particles. So this is really beautiful. If you have particles with different sizes, we can cluster them according to the flow field, because bigger particles will not necessarily follow the shear flows as well as the smaller particles, which means you can use these in your favor to separate things at the microscale. So this gives you now a tool from which mathematics can be used to control or have self-organization. And this is the last example in robotics, where now this is much bigger scale, and we try to emulate intelligence, or what is called in robotics embodied intelligence. This terminology comes from the fact that you don't need controllers to control your system. Emergence comes from the interaction of whatever you have there with your body, and your body has to be soft, has to be an elastic body. If you make it rigid connectors, then you go to the more traditional robotic system, which you have like cogs and rigid parts, which I think is a part of robotics. Is beautiful, but I don't think this is going to be the future, because no intelligence, like you have to code intelligence there. Whereas if you have softness and interaction between parties, then you can get around without the need to control the object. You can get around by exploiting these emergent properties. So, for example, this is a oops.
Double that screen.
Yeah.
Just in the middle. On your flat. Just double tap on the screen. Yeah. Just in the middle. In the middle. Double tap. Double. Yeah.
Okay, then goes to full screen. Cool. So, for example, this is an example that you have an object with suction cups. Each suction cup has its own like a pressure supply, but it has a hole. So as the cup, as one hole is sealed, increase the pressure in the structure and then bends. The other hole is sealed. So you create a zipping effect that you don't have to code it. It's an emergent property. And that's what biology keeps using. You don't need to devote a neuron to this kind of task. You can just do it without having to record your hardwiring control, is what I mean. You could make a robot that will have, that will do the same job, but that's kind of cheating, because you get what you put. Is usually what I say. Like, if you get what you put, that's not usually the game. Nothing against. Sorry if I'm nothing against, but that's a little bit outside of the scope that I like to study. So, but I'll not be talking about these things. I just want to give a little bit of an idea where I came from. So back in Brazil, I started my career as a scientist looking at these problems. This is the so-called moving boundary problems. So you have two fluids with an interface, and the interface evolves. At the time, a lot of mathematicians and physicists were really interested in this problem, because moving boundary problems usually have a PDE where the boundary condition has to be satisfied at the boundary, but you don't know where the boundary is. So the boundary is an unknown in itself. And as a mathematical problem, this is hard. However, this problem in particular that we call this, if you have a less viscous fluid injecting in a more viscous fluid, this creates a fingering instability. That's called Sattman-Taylor instability. And this problem you can get around with second order kind of perturbative mathematics, surprisingly well. And then because of that, even though this is highly nonlinear problem, because of that, a lot of mathematicians went crazy in that period, working in this problem. And many people that today work in maths bio, that maybe or soft matter, like of live things and live cells and things like that, they actually came a lot from this field, which I feel And I kind of continue this through my life because every time I see biological system, they, in one way or the other, cells moving inside your body, they have a membrane. If it's deformable, that means already that is a moving boundary like this. But the boundary is elastic and then it's interacting with the fluid outside. So you have multi-physics as well. You can have chemistry, physics, and other types of interactions, like it depends on, but then the coincidence is that one of the people that invented the device is called Hilary Shaw. He was the first professor in engineering in Bristol. So to me, it's special because when I left Brazil, I studied only that, and I never saw any experiment of that. And then to be able to somehow life take you to a place where you started your the topic that you started, I found a really beautiful symmetry for me in particular. So since that time, I kind of I try to see a trend in the kind of things I do, and I think the trend is I like to work on things that have some form of visual beauty. I think I'm naturally drawn for things that have patterns. Patterns, either in time space, or like in here, for example, you have these tip splitting or radial types of patterns. Like this is like beautiful in many ways, So I think this is what draws me. But I think for almost good 20 years, I've been studying a lot this kind of mechanism. And this is what really I want to talk today. Yes.
So just regarding your last slide, so is there like a condition that, like in the literature, is there a condition, like general condition that needs to be satisfied so that one sees this kind of instability, or like what do people look for in these problems in this literature?
Hmm.
Is
I'm
there,
not sure if I understood your question. You want to know where?
do we know a condition where like which each of the sides of the boundary might satisfy, or like certain condition under which we can see this kind of
Oh, yes, absolutely. So, for example, there is when you do even linear instability analysis, you see that your growth rate is going to be dependent on some parameters. One of these parameters is what we call the viscous contrast. And for you to have instability, you need the viscous contrast to be from, I'm making a mess. From low viscosity to high viscosity. If you invert that, if you inject the fluid that is more viscous in a less viscous, that's a stabilizing situation, unless if you press really quickly. So for example, these linear instability analysis already tells you the growth rate will be either positive or negative, depending on this parameter. And that's why it's so wonderful in that sense, because to answer this question, I don't need to even go to higher order in the expansions of this problem. And there are many systems. We are interested on the pattern itself, but most of the time industry wants to suppress the pattern. So I had many colleagues where, especially spin coating, you need to do spin coating for your driver, your CDs, your DVDs, not anymore, but like, let's say your drivers, or if you bake in a microchip, you need to put a fluid and spin, and as you spin, you don't want these instabilities to arise. So for example, this is the spinning, this is the type of spinning problem. A droplet, oh no, I did it again. A droplet that is in the middle and you rotate. So you have the centrifugal forces that are pulling this droplet forward, outward. But this situation is what people don't want. So what they try to put is like more viscous inside, less viscous outside. And air counts as a less viscous environment, for example. But then if you suppress, I lose my job. Because then I can't write about, in the papers. What we care is, okay, when the stability happens, okay, what kind of stability you get? Industry wants to know, when the stability get, I will avoid all this together. So it's a funny like relation with if you want to work with stakeholders, real stakeholders. So for example, this is oh, there you go. This is the situation of a rotating helical cell. And this is the situation if you have a Coriolis force, so inertial effects, for example. And then you see the fingers start to bend in one direction, opposite to the rotation. So that's why you see a slight bend of the fingers. But to see this, because this is such a highly nonlinear stage, an advanced stage of the dynamics, you have to really simulate this for a long time. But the system that I work these days is this one. I can spend hours just looking at this. So this is a sperm cell. It's a sea urchin sperm cell that is swimming in seawater. And you are here in the frame of reference that moves with the cell. So this is a free cell, but all the images have been overlapped so that you can see the frame of reference that moves with the cell. And then you see these beautiful traveling waves that emerge. These cells are about 50 microns. So you have molecular motors that are deeply embedded there that are doing something. Also, this has to swim, but not just swim. It needs to find the egg. Once it finds the egg, then you have reproduction. So yeah, we wouldn't be here without that, right? So suffice it to say, this is very important. But this is one of the most primordial muscle units. Because in here you have self-emergence. If I chop this head, this thing can self-oscillate like this by itself, by isolation. So I don't need again neurons and things to control this. Self-emergence occurs in isolation, and you have a physical system that these will always be true. If you put all these ingredients, you have the waving so much that you see this kind of strategy everywhere in nature, everywhere, including our bodies. Our bodies, every organ has a type of cilia that is responsible for beating flows at that particular organ. If the cilia is defective in that organ, you will have one of the so-called ciliopathies. So it's a disease associated with a defective cilia. And because every organ you have a cilia, you have diseases that affect every organ. Your eyes, your brain, your lungs, everywhere. And sperm for the male population, of course. And often if you have a problem of defective cilia in, let's say, if you have chronic bronchitis, if you're a man, it's likely to be also infertile, or that will carry these so there's a lot of importance from developmental biology and evolution as well. But I come from the perspective of how does this take place? What's the minimal system that these could be allowed? And yeah, how self-emergence occurs in that particular system. And also like if they need to swim and find that, how do you make this decision? You are a cell, you don't have a brain. How do you make such important decision? I'll give you an example. In some species of fish, they only have less than 1 minute to find the egg, otherwise they'll die from osmotic shock. And they are released in open water. So you are at the micro scale, and you need to really quickly find the egg, otherwise that's what's the point in life. So they are very good at finding maximums and especially moving through gradients of chemicals, exactly. So the expert, optimizers, if you think from that perspective. So the dream that like I have is that one day that we as a community will be able to find the governing laws that uh, can describe this system properly. So to just give you a little bit of background, you have the genetic payload here. This structure is complex. It looks like an engineering structure. So you have a bunch of fibers we call microtubule doublets. They are connected by elastic springs, radial spokes. There is a double band here. This geometry is also conserved across the species. We call this the 9+2 axonym. 9 because you have 9 outer filaments and a pair in the middle. And you see this everywhere, in your lungs or in a sea urchin sperm. It doesn't matter. You see, like, this is evolutionary, like nature found this solution and everybody uses. Doesn't matter if you related or not. For example, with sharks, you see the same structure, you know, and that's also fascinating to see. We don't know about that, how it takes place. And then you have molecular motors that are deeply embedded here, different types of molecular motors. They exchange chemical energy to produce work. So, and they produce work by really moving parts. If you have two filaments, they will try to shear these filaments, because they are interconnected, shearing motion is then converted in bending motion. Beautiful. But the original motion is shearing. And then this bending motion is transferred to all this structure. And then you have chemistry that couples with mechanics, and then there are also couples with hydrodynamics outside. Really complicated problem. And there are many questions, and I work a lot in all these different interactions. So at the molecular motor chemistry level, so reaction kinetics, at the mechanics level, so how solid mechanics of filaments, cylinder bodies, and things like work, and also at the fluid level and their interconnections. So you can choose the game you play according to what you like, but to encompass everything, that's really, it's still really, really hard today. Very few people, I think, maybe have the courage to do this. A lot of people in this, but like connecting this is more complicated. So I'll talk a little bit about how we go on these interconnections and how this path is really like is a winding path, in the sense that everything that every finding that we stumble upon kind of like is a really small iteration for the next iteration. So I'll try to show a little bit about this. So for example, because they are really slender, and remember I showed you the picture, there's all this structure inside, but when you bend, you just see the bend, right? So for a long time, you see this bending, this whole structure bending, and as a reasonable approximation, you say, okay, this bends like an Bernoulli rod. So a simple rod that the bending moment is proportional to curvature. Okay? The Bernoulli rod says that. The problem is that in some experiments, in 2005 and 2009, people that disabled this motor, so the structure now is free from activity, is dead. And then the head is stuck to the substrate, so it's pinned. And then you come with a microprobe and you bend this structure. Then, if you bend this structure locally, this should just go straight because this part is free. It's a tip free from torques. But if you bend here, this outer part bends in the opposite direction. And that was thought to be paradoxical, because you have a counter bend, but you don't have any forces there, external forces. So the conclusion was that somehow you have some internal forces that can be accumulated and transferred to this point so that it bends itself, which is really weird as well. If you think about the material that is non-linear that way, that's really weird. And then because rat sperm is complicated, it's mammalian cell, etc., they done with sea urchin sperm, in which the structure of this filament bundle is way less complicated. And they found exactly the same, not exactly, but the same phenomenon happening. And in that, for a long time, remained like a big puzzle. Like what, a lot of theories, a lot of ideas people put forward, but no mathematical model that could actually connect the observation with these ideas or theory or hypotheses that were at the time. And this is the whole elastic equation. And if you solve with point forces, for example, here, and you're also solving for this this part, so this is actually a solution, it's not just a drawing. You see that nothing happens here as it should, because curvature is proportional to bending moment is proportional to curvature. If bending moment is 0 here, external, curvature should be 0. So you can understand already from boundary condition perspective. Yep. So at least we established that it is definitely not an early Bernoulli rod, which is bad because a lot of theories use early Bernoulli rod as the so we have to revisit these results. And then you can come with a naive way and say, okay, this is a filament bundle. Let's make 2 filaments, connect with elastic spring, and solve the PD of this connection. And also you don't get the counterband. You do get a little bit of slide in between, but you can see that, so this is the amount of sliding that you would get here. But because I'm bending this and I'm bending this in the fluid, we don't get the counterband. So it's not as simple as just connecting elastic spring. There's something else in this structure that is driving this. And in this particular example, I have just elastic springs. But then if you actually, I thought I have a slide with equations at the end. If someone has questions, I can present that. But the good news is that if you couple these filaments in non-locally, we had a big discussion about what locality is. So, but if you couple these filaments non-locally, in the sense that the bending moment now is not just proportional to the curvature, but the bending moment depends on the accumulated curvature of the whole system, then you have this excess of bending moment that is generated internally that then bends this part in the opposite direction. So this comes naturally from the model. And because this section here is a linear equation, you can solve for analytically, and then you can fit with the data.
Sorry, then compared to the previous picture, it means that you have cross links between the filaments, not only parallel. Like
I have accumulated bending moment. So in addition to the cross links itself resist bending, not just extension. So this model embodies that. It's not just linear extension. The links, they resist bending on them. If you think them as they're connected in each hinge point, they are not hinged. They're kind of clumped between each segment. And then you get a bending moment associated to that. And because of that, then you can use this solution to fit the parameters, and these parameters, they embody the bending stiffness, the shearing stiffness, and also the basal body. So at where the filaments meet here, they also can slide, and this has a very complicated non-local effect in the far part of the system. So no locality in terms of this. So we solved, I'm not going to show, yeah.
Not to be very critical, but we did this one time. So you had the actual model, or the Bernoulli model, this didn't work. And then you added an extra mathematical term.
Yeah.
And this extra mathematical term solved your problem. And then you have.
No.
Can you now go back and sort of point at somewhere in the like whatever.
No, it's not. Yeah, it's because I needed more equations to give an idea. Before we came with this model, let me see if I have. I don't have the equations here. I'm afraid. No. Yeah, I'll have to go back. So to answer your question, the answer was not as simple because at the time, what was understood is that there is some form of shearability in the system that people are not taking into account. And null theories, they are not shearable. You will start from the perspective that when you bend, you have such a slender body that shearability can be discounted. But then there are many schools of shearability. You have Timoshenko, you have Egenser, you have like, God knows, And I studied all these types of shears, the Coursera shear. In none of these shears, they were able to provide the right answer. And the reason why is because they are local shearing models. Shearability will only happen locally with the curvature coupling. This is a model that embodies shearability across the whole system. So to answer your question, it's not that one model without a model like Autonoia. There was a progression, there was an evolution, and this is also based from bottom-up approach. So we are not, is different from when you do mathematical biology, that you have a data set and you come with a more phenomenological parameters and you try to fit. Here, everything is like bottom up. So we add effects as they are needed or reduce as we see fit as well. So this has a physical groundings.
What's the physical intuition for why it would be typical of the curvature of that form?
So the physical intuition goes back to the idea of the workings of this structure itself, because as one filament slides past the other and they are coupled by elastic springs, if you see here, these are filaments. They are not extensible filaments. They are like a rigid filament. So if you think about a railway system that you can just slide, this is going to be transferred to all other neighbors that you have this similar displacement because of inextensibility on the filament. As a matter of fact, what I showed is that this non-local theory can go back to the local theories like Coursera shear if you make the filaments extensible. That's right. So the way of non-locality is coming because the filaments themselves are not squishable.
There's like a Lagrange multiplier.
You have a Lagrange multiplier, yes. You have a Lagrange multiplier on the fact that the filament, you want to conserve length as much as possible. Of course, this isn't unreal. No filament is truly inextensible. But even if you add a little bit of extensibility, you can still have this phenomenon.
Is it analog to this Gauss theorem, electromagnetic integration onto what you?
It could be. The problem when it comes to continuum mechanics is that when you like there are many complicated materials that we don't know about the constitutional form that they have. So I agree that you could have a competing model with a different setup that could give it a good fit as well. But that's not the problem with the theory. It's just like there might be many possibilities that you can get the same effect. Now, another question is model identifiability and whether a model is right or not. So even today, if you have two competing models that both fit the data and all the physics or everything that people know at that particular point, there's nothing that makes you really choose one against the other. So what we do, we use the Rosen razor and say the model that is the simplest should be the one that should be used to explain the data. But even this is like a big idea. There's no proof, you know. So because this is simplest, but I agree, we could generate a family of models in which, and this problem of model connection with data will come again in a bigger problem that I'm going to show. This is just like the preliminary stuff. The good complicated stuff will come when you have the dynamics.
Would it be possible to build with the deal using just strings and text or Lego, whatever, and just see it happening with your eyes? Or
Yeah, that's the thing. Like we're trying to do this in the lab, but this is incredibly hard because because there are many elements connected. But this is also the good thing as well, because there are so many parts in this structure, and we're trying to make sense of what are the essential parts, what are the parts that we can throw away. So I'll give you an example that in this field is no longer acceptable to use Euler Bernoulli theory. And if you do, you need to really come with a really strong idea, because you will not be capturing something that is happening at the larger scale. So remember, this sliding is happening almost at the nanometric level. The accumulation is having an effect at the microscopic level. So it's no longer true that because it's small, doesn't have this, because when you go to the limit of small deformation for this, they're really truly small. But this is amplified. And that's for me the dichotomy. And that's why some of these problems, they remain unresolved for decades. This one was 30 years, almost 30 years. So, so for example, now that we know a little bit about this, how about the molecular motors? And how about also we couple the fluid mechanics? Because these were just mechanics, pure mechanics, without any of these other stuff that I mentioned to you.
You have similar effects on
Twists, yeah, they're coupled twisted, yeah, because sliding is coupled with both the curvature and the twist.
And does the model also take into account the extensibility condition on the bending stiffness as well?
In the 3D models, yes. At the end of the talk, if I have time, if you guys are not too tired or too hungry, I can brush it onto them as well. So then you need to solve this problem. But of course, this is very difficult, because now you're talking about solving Navier-Stokes equation, and then solving Corsair rod theory, and then solving chemical reactions. Like, suffice it to say, there is no solver that can cope with that, like a commercial solver. As a mathematician, you have to build your things yourself. And I have many methods for these, and for the integration of the three. Took years, like really years, to find a method that would cope with the problem that I'm going to show you. And it's just a really slow progress. So now I'm going to introduce the reaction terms that comes from the molecular motor, which is very similar to what you find in your muscles, myosins as well. So the motors here are different. They are called dyneins, but myosins, kinesins, and they operate in very much similar ways. So the way we model them is like teams of motors that they will have either two states. Either they are attached and producing force, or they are detached. And they have a chemical rates for attachment, chemical rates for the detachment. The mechanical coupling comes from from the chemical rate of detachment. Because the more force the motor feels from the system, the bigger is the probability that it will detach. And this comes with the exponential of the force. So you use this from Kramer's equation in like a reaction chemistry. And the probability of attachment is proportional in this, to simplify everything, is proportional to the number of motors that are available. If you're detached, then there is a great probability you'll be attaching again. And then you need to couple this with the filament bundle system that I mentioned, that is non-local in terms of these integrals. The problem is that the equations are really complicated. And at the time, we tried everything, and we didn't manage to solve this numerically as well. So we could do a great deal of the linear instability analysis, but to solve the full problem. But at that level, what I was interested is this type of simple idea that have been put forward before, but never solved at the nonlinear level. So that was just an idea. Would these work or not? In terms of, will these simple building blocks lead to self-emergence that saturate modes or not? And what I was interested was not on the nonlinearities that come from the structure, but the nonlinearities that come from the reactions, and see if they are enough to make the system saturate. So here you have the fluid structure interaction equation coupled with the chemical reaction from the molecular motors. But this is truncated in the small curvature approximation. So here I don't have the Lagrange multipliers, for example. I'm not solving for large deformation. I'm just solving for a small curvature. You could have slightly large deflections as long as the curvature is small. And then what we found is that the system can saturate. The motors self-organize in two teams that oscillate, and you have a traveling wave that emerged without me saying any qualities of that. And the more you go away from your linear, from the bifurcation point, so this is a Hopf bifurcation, and this is measuring distance from your Hopf bifurcation, then you get, of course, more nonlinear, more nonlinear. So this kind of showed us this was a good model in the sense that I could have saturation of modes that was bottom up. So I'm not trying to come with phenomenological structures. This is comes from the chemistry and etc. The problem was that the waves that we get, you saw the wave that I got, right? This is not really like a sea urchin sperm. And big part of my work as a modeler is always like, I could spend a lifetime doing pretty models. That doesn't mean anything. It means to me, I got my grants, I got my papers, but then what's the point, you know? As a modeler, a big part of me and big worry is how this connects back to expression. This is really big worry, constant. So at the time, I abandoned this model by just looking at this. I saw this. This doesn't look like anything I ever analyzed in my entire life as a traveling wave. Therefore, this model is wrong. And I discussed this in the paper. I discussed this. And I abandoned this model, and I went to research other things, other models, and see. In the meantime, colleagues in US took this model and managed to solve at the nonlinear level. So better numerical analyst than I am, because I'm not. And they actually showed that the traveling waves you can get traveling waves that are really nonlinear. And if you look at their papers, you think, oh my God, maybe this, after all, is not so bad at all. Maybe I should rethink. And the problem is that in here, I'm constrained because we solved for small curvatures. So maybe I was asking too much from the model, you know. As soon as you go to monolinear stuff, then you get more realistic looking waves, which is an important proxy, because then the work to compare with data is so much that you need to have some kind of early, like early symptom that is worth going that direction. Otherwise, you're just going to spend time in not get anywhere. So I came back to revisit this, and this was also work with my PhD students. And now, and together with all these people here, so they have their independent paper, and there are many more that use our model in their system to make emergent behavior like this. And these are the type of waves that we get with this simple. Now solving the elastohydrodynamic problem in the nonlinear sense. So this is not constrained anymore to the fact that I have a small curvature. And then you do get, so this is like a simulating chlamydomonas, this is like simulating sperm cells. What is important is that all these different traveling waves are not encoding them. I set parameters, they self-organize, you have a transient self-organized, the mode saturates, and this is the emergent behavior. I don't have any control whatsoever on that.
You have parameters those to the reaction, the molecular models, plus the...
Yeah, exactly. So you copy the whole thing. The fluid, the elasticity, that is non-local because of the sliding moments, and the chemical reactions.
What do we mean if we don't set the parameter and we put it in the computers?
No, it's because this makes the difference between many people that solve the fluid mechanic problem. As I mentioned to you, there are many questions in the same object. So, for example, I could come here and I'll show you some of these examples. Here, and this is experimenting that we've done a long time ago. You see how sperm moves. You track the cell, you see the shape. So with the shape, now this means you can fully solve the hydrodynamic problem by imposing the shape. Because it's a moving boundary problem, so you basically say, whatever the shape goes, enforce no-slip boundary condition there. And then you resolve for all the forces, for all the torques, and the fluids flow around, which is something I will be showing later. And this is, for example, one measurement, if you wish, of the fluid flow of this particular cell. So in here, I didn't have to do any of that mass because I'm imposing the deformation. What I mean is that the waveform, and this is very important, the waveform you get is a result of everything.
What do you mean, like,
You're not imposing, you're not like because you have to make a distinction between models where we'll come and say, okay, my model is that my wave is going to be exactly like this. And then you solve the whole fluid mechanics. But then maybe your question is more about what happens if you have many swimmers swimming together and they collect the swarming behavior.
I think my question was, maybe it's just semantics, but when you say that you don't set parameters and this...
No, I said we set the parameters and you get the waveform as a result of these interactions.
And you call it emergent.
I think that there's a vocabulary that probably what you mean is your whole thing is mechanistic, and of course there will be three parameters from the mechanism models, and that can produce the behavior. Whereas when you separate these three frameworks, you can constrain for a specific framework. But then in the end of the day, you need to fit.
Yeah, exactly.
Yeah.
Yeah. So for example, to get a particular waveform, you have to say how your motors are doing, what's the viscosity, what's the elasticity, and then you get a particular waveform. Maybe what I'm failing to say is that when you look at experimental observation, there is a huge diversity of behavior that you see there. Huge diversity of behavior. Because they are mechano transducers, these things. They will react to what is in the environment. So and the reason why is because these molecular motors, they are chemical, but also they are mechanical. So if the fluid structure outside, if the fluid increases the viscosity, they self-organize in a completely different way. And here's the example. This is human sperm cell in low viscosity, and this is human sperm cell in high viscosity. And to this date, there's not an answer on how this is actually taking place. Because it's an emerging property of everything that you have. And if I change again the conditions, especially the physical conditions, there will be another solution for this waveform. So this is a big question, is that you're trying to find out this from bottom-up approach, not top-down, because I could start with a model, in high viscosity, this happens, but then you get what you put. That's what I'm trying to say. Here, I'm doing bottom-up. I don't get what I put necessarily, so much that in the early iteration of this model, I thought I was wrong, and I completely abandoned, to be honest. If you compare with data.
So you show that we get a waveform for two people with parameters, and then if you go in a different viscosity, you get a different waveform. Is there one steady state, right? If you even within one viscosity, you perturb it very far away, you always come back to that same waveform.
So steady state, because they're all cycling through a half-life location. This is the steady state.
Yeah.
So you start with initial conditions, and that's another important thing. The solutions I'm showing, they are steady state solutions after they pass through a transient, and they stabilize into this solution, and we can say it's steady state.
Regardless of initial conditions.
Regardless of initial conditions, yeah, exactly. So, and
There are two possible websites.
yeah, definitely could be. And also you have a region in perimeter space. It's a half bifurcation where no oscillation occurs. And this is also very important for us because then we can go to the experiments and interrogate if cells will be actually moving and being able to self-organize in this perimeter region. So is a model that can be fully tested with or without the motion in that sense as well. But it's still very hard because when you mess with biology, you mess with everything. It's very difficult to say, oh, I'm going to change this parameter, but has a knock-on effect in so many other things. It's like a proper complexity. So if I go, but then the problem that we had was that, as I said, I wanted to compare these with experiments, and yeah, and having the three interactions were too complicated. So how about we drop one interaction? And because there is a lot of work in fluid mechanics, then how about we kind of see what happens if we drop fluid mechanics? We have reasons to do that. Okay? So I'm kind of inverting we have physical reasons and mathematical reasons how to do that. I'm going to show in 1 second. But what happens if I remove altogether my complicated fluid mechanics and I stay with my mechanics of the axoneme itself and just the coupling with the motor chemistry, but without simplifying or constraining to small amplitudes. So you keep the system as it is. You just reduce the physics. You don't reduce the complexity of the model after that. So one really interesting thing happened because when you look at this system, this brings usually reaction terms, so chemical reaction from reaction kinetics. And this brings a diffusion term, diffusion-like term, because bending moment is proportional to curvature. When you go to bending moment density, you have another derivative there. So this gives you a second derivative in the center line or in your tangent angle or whatever is your proxy for deformation. So this is in essence is a diffusion term. So much that if you bend one structure and if you release it, this structure is going to unbend to the straight configuration. So, and the speed in which it unbends is the same speed as diffusion. But I'm talking about diffusion, not of molecules, diffusion of bending, diffusion of deformation. So diffusion is here of the deformation, not of molecule. So I have diffusion of deformation with chemical reactions. And the system of equations are much simpler because now this is the structure. You have the time here. You have the diffusion term that I mentioned. This is just because of the curvature. This is Euler Bernoulli diffusion. Euler elásticas are nothing more, if you heard of, are nothing more than the diffusion equation in the steady state for rods, for the formation of rods, right? But then here you have the dynamical problem where this is coupled with reactions. And these reactions, they come from a bottom-up model that's telling you how the molecular motors are interacting with this structure. And then what we get is something like a Turing pattern. It's not a Turing pattern, but it is pattern here. It's not pattern of visual patterning. It's patterning of motion. And patterning of motion is something that happens in time space. So for example, one common solution for this problem is a pattern that is stripe patterns that you see also in reaction diffusion system. But these stripe patterns, because they are in time space, they are bending waves that are traveling along the tail and on the course of time. So these are waves of curvature that are taking place. And this is a common solution that you get from this. So now we have a very simple system where basically three equations. You can find the code in the paper as well. Three equations where you can simulate that will give you a reaction diffusion system, but no classical, because I'm talking about animation, not diffusion of molecule. Also, remind you that the reactions, molecular motors are not diffusing around as well. So you don't have motion of the reactants. The reactants are stuck in the structure. So you have reaction only, although you could have a diffusion term of here as well if you want to make your thing more complicated, but you don't need it. So you put these things together and you do get these traveling waves, and they're very robust. Then the next job that you can think of is like, let's look at experiments that people have done on this. And let's try, now that you reduce the model to the bare bones, can we try to see if this can fit data, real data? And this data is complicated because, again, as I said, it's time space. So it's spatial and temporal. And this is an example of experiments that were done in a lab in Germany by Howard, and that is actually in US, but these experiments were done in Germany. And these are simulations. But then we can move on to something more quantitative and actually fit the model that is now nonlinear for the first time with the data. And these are the fitting results that you get from Chlamydomonas is this type of microorganism that has two flagella. And you have a mutant that the beating is symmetric, and the wild type is asymmetric. So you always see the beating doing like a breaststroke. This is a mutant that have a symmetric mode always. And these are sperm cells, bull sperm cells. These guys, Chlamydomonas, they are about 20 microns, so they have very short flagella, 10 microns flagella. Bull sperm goes all the way to 50 microns, so much longer. And until that, the theory and the field was divided because some models fitted well with this, other models fitted well with this. But we didn't have one model that could join these two things, so much that until we published this paper, we had different ideas about how the control is happening at the molecular motor level. So now you have something that is very parsimonious, as a model that is really simple to the bare bones, that can actually connect microorganisms and cilia in different scales. So one of the conclusions is, if the model is capturing really well this also means that many aspects of the model should be correct, including the fact that we remove the fluid mechanics, which is really weird, because the fluid is the sole reason why these things beat in the first place.
And then how do you reconciliate with the slide that you show that the density change the wave? The viscosity change the wave. You show this slide, two experiments, different waves in different densities.
Yeah, I'll show you, the problem is that because we reduced in this model, this only, let me just explain one thing, and then I'll come to this comment, because otherwise I'll have to invert the, but I'll keep your question in mind, and this is a good example of what was the state of the art, for example, for chlamydomonas, and trying to fit with a particular model, but the fits were really, if you see the curves, that's not. And then when you go to the nonlinear level, then it gets much better. But with just these three equations. So this gave us confidence that maybe this model is capturing some aspects that are right, to the point that then we can interrogate, is it really true that we can drop fluid mechanics? Because from the perspective of the complex system, they evolved so that they can move fluid. So how is that possible that fluid is completely invisible to them? If I turn off fluid mechanics, they don't need the fluid to self-organize, right? So now we have an apparent paradox here. And then to resolve this, we put back the fluid mechanics and solve the full system. So this is the full system with different motor activity and increasing the viscosity here, for example.
That's what the...
And that was your question. And we wanted to say how confident we are that this is capturing the system. And basically, you have, let me see, next equation is better. So this is the full system. Sorry, this is tiny. And these are the equations. So these are the terms that you need in the equation. So you have the molecular motors, the elastic bending, and then the viscous dissipation, for example. So then from this, what you can do is okay, I have my full model, I have my simplified model. Then you literally just compare one solution with the other and see how close this is first. And then we find that there is a decoupling region from that. But then you can go further. In this decoupling region, let's measure what's the amount of viscous force that the filament is actually getting. And this is the pink curve that you can see. Oops. The pink curve that you can see there, that is very, very small compared to all the other forces that are in the system. So indeed, in this limit, when you are in low viscosity, so for aquatic species, all these species that have to swim, marine species either in the sea or in rivers that populate our planet, they are in low viscosity. They have to swim, they have to perform in low viscosity, and they don't see the fluid. They don't perceive the fluid. The molecular motors are simply washing through. The fluid resistance is not enough so that they can feel the feedback from the system, from the fluid. So it's almost like they are in a different space, really, in a different space. So that gave us confidence that maybe this model could be used, but only confined to this regime. Because, of course, as you increase the viscosity, then the viscosity starts to become important. And of course, this breaks the validity of the model. So the model is only valid for the marine, small small viscosity regime. But also this is we think that the validity is more because there is one aspect that we didn't include, and here is the internal viscosity. So we're not including the internal viscosity of the cytoplasm of the cell itself. Because if you add this, you have another friction term in the system. That means that the molecular motors will be even less sensitive to the external viscosity. So we think that here is where it stops the validity, but we think that effectively is going to be larger than that. This is like a very cautious regime that we think the validity of that.
You're talking about high and low viscosity, but I'm just trying to remember last time.
You're trying to
I'm trying to remember still from my second year course on this kind of how bacteria swim and these kind of things. Isn't this something more related to the renal? Where like you have to storage, even sort of in a almost zero.
No, everything here is happening at low Reynolds number still. Because Reynolds number only distinguishes where you have inertial effects or no inertial effects.
Okay.
But once you don't have inertial effects, you still have effects of viscosity because of the elasticity. If you're dealing with a problem that is just in the fluid mechanics perspective, viscosity is just a parameter and you can non-dimensionalize and take away. But because you have a fluid structure interaction, then the bending and viscosity, they enter in a non-dimensional group. So much we can have this number that we call sperm number is the external viscosity, but also is the bending stiffness. So increasing this number is equivalent of increasing viscosity or reducing stiffness, for example, and vice versa. It doesn't matter. From the eyes of the equation, it doesn't matter because they enter in the same dimensional group. So usually I use here viscosity because this is what people are more interested in, what the kind of thing that they can change in an experiment. It will be hard for you to come with a mutation, choose a sperm cell. Imagine you need to mutate a human so that it produces a sperm cell that will have a different bending rigidity. That's not going to happen. Even with mouse models and things like that. So what's the parameters we have to play at external viscosity? So, and that means that as long as you are away from these, you can say that the reaction diffusion is the central core of patterning the system. And that perhaps what happens, from the Alan Turing perspective, since the seminal work, the community have been in so many directions, hoping that maybe reaction diffusion system could be mapped into biological systems, but they are hard to find. You have a lot of reaction diffusion papers everywhere. You have maybe 5 papers where we will show that reaction diffusion can be matched with morphogenetics, and we believe that maybe this system is one of them as well. But this is non-traditional in the sense that we're not talking about diffusion of molecules, it's diffusion of bending. And the reactions are also not moving with this, in space. The reactions are localized in space.
But if I heard you correctly, you said that the diffusion term in your bending comes from the Bernoulli approximation.
Yeah, from the curvature term.
Yeah, but the first part of the talk was trying to convince us that your Bernoulli was not the right.
Yeah, no, fair enough.
Think about.
But it's because the non-local theory has the local, which is the Euler-Bernoulli theory, plus the non-local term. So you still have Euler-Bernoulli there to give these local effects, plus, and this is embedded in the constitutive relation of the structure. And this is embedded in here as well. Yes. So So I'm trying to be consistent with the things that I think are important. So much that something that I hypothesized early in my career, and only now I'm being able to actually interrogate this question, is like, since I've been working on the contraband phenomenon, for me it was kind of obvious that if you have contraband happening, there's a quicker way for motors to self-organize. Because if you have teams of motors at the beginning that are able to synchronize, they'll send this signal non-local already to the others to be out of phase so that you have a traveling wave. And that's a really clever way to have emergence behavior. But people think more that molecular motors are the clever entities. I think more that the structure is the clever entity. The structure is what is organizing the motors. The motors are stupid. The structure is in such a way that organized, and I will have another example at the end with a robotic example like of that. Well, at the end of the day, these are all ideas difficult to prove or disprove, but it's a game you can play. So now this means you can solve a completely different problem in here, because if fluid mechanics is not important, then you can solve this problem in two parts. The shaping problem, where fluid mechanics is not important, so you solve what the shape is going to be. Once you know the shape, then you go to your fluid model to solve how this shape will make this swimmer swim, which is interesting. So it's not the hydrodynamics is not important. Without the hydrodynamics, they don't swim. They don't move fluid. But hydrodynamics is not important in shaping the beat, in manufacturing the shape. And that for me was hard to come to terms with that, because I started my career as a fluid dynamicist. I came from Brazil, and fluid mechanics was always more central than anything else. So for me, it was really hard to come to terms to this. To then come and say everything that I worked, it's not that it's wrong, but it was unnecessary, And at the end, I could just put this aside. Of course, if you need to go to the full problem with the viscosity, you still need hydrodynamics. And that's the
One question. So basically you have a boundary where hydrodynamics is needless and all
in
beyond
the
it.
parameter space, yeah.
Yeah, in the parameter space where then you do need it experimentally, where most experiments.
Yeah. So it depends on the species, because many people are interested in marine species and they are in water, seawater, then low viscosity is more important. But because I also work with bodies and things, the viscosity inside our body, like sperm has to move through cervical mucus. It's a highly viscous, highly nonlinear viscous. So I can, if I want to understand emergence there, I need to add all this. So is a solution that is only for probably what we have more common in the planet. For sure, more marine species than anything that is. But also you have really funny species like sharks. They evolve as internal fertilizers. So whenever you have internal fertilizers, they have the axoneme and more structures around them. Then they went to be external fertilizers. So they put sperm and egg, outside the body, and these drives things in a different way. And then they came back to be internal fertilizers again. So you have some species where they have some sense of what low viscosity is like to be, evolutionarily speaking, and then coming back to internal fertilizer, where fertilization is happening in an enclosed environment. But the price you pay is that the complexity you have from the environment, like viscous. Viscous is really hard. So one thing I didn't say is that most of these systems, you have 100 times more viscosity than water. 100 times. Oh, I need to finish. So, but before, let me just, I have a few things. Let me just, so now you can recast this system in a reaction diffusion type of system. So here is like just your diffusion matrix, your linear operator, and then your non-linear operator. And you can play the same business that people do with classical reaction diffusion, but here you're getting patterns of motion, which is quite cool. But one thing I wanted to show is a little bit of what happens when things get more complicated. So I was showing things that were in the plane and also things that were in low viscosity. For these, we needed to come with a completely new method on how to solve the full elasticity, the full fluid mechanics, and the full motor chemistry. So we devised this model and implemented to 3D now axons. So now I'm solving everything, and also you have the 9 filaments, and each of these pairs, they will have molecular motors undergoing the same chemical reaction. And what we find are patterns that are emerging, not traveling along the length, but also spiraling around the tail. So if you unwrap this is what you would see. And this is the free swimming motion of this. And for example, we can test some ideas that we find in biology. This structure is conserved, but in some species you'll find a breach that is stronger than other species. And people hypothesize that this breach is what makes the beating planar versus a beating that is helical. This was a hypothesis. So we came to test this hypothesis. So this is something, an axonym that is isotropic, if you wish. And then as you increase the stiffness of one breach, then you get the planar switch, in which now motors, even though the motors are, and again, this is self-organized. This is emergent. I'm not imposing anything. The motors self-organize into 2 teams, even though you only have one blockage there. There's 2 teams that self-organize, and that switch in a coordinated fashion so that you have the planar beating. So we can ask questions about, and this is what is happening. You see the wave going around the axonym, and here the wave now is stabilized in 2 teams. But the boundary is here, if you wish. The stiffness is here. So we now have been able to interrogate things in structural biology that people say, whether in this model we could see a resonance. Now remember, I'm very concerned on how we then go and move to data. The data is complicated. Like how is virtually impossible for people to image molecular motors at the nanometric level, and then also have the information at the macroscopic level while everything is rotating and running. So what people do instead is a fast freezing, and they do cryo-EM of many cells. And this is a result in which people try to say which motors are attached, which motors are not attached in different sections of the axonym. And then we can go in the model and say, is this happening in this particular timestamp? So this is a more qualitative comparison, but to get quantitative comparison here, at the moment, we reached a point where that's not possible. But so far, this looks very promising. And then, for example, things like synchronization. In chlamydomonas, one of the bigger problems is that we have these 2 cilia, and there are no transients really. If you change the condition, it will change the beat in one way. That is, they beat with 60 hertz, so you need at least 1,000 frames per second to record this motion. And the way that they change it, they go back, is almost like they have no transit. But when I go to the model and I see the computational time that it takes, especially when you are in low viscosity, so this is the low viscosity regime, the computational time that it takes for them to go to this steady state is huge. How these 2 filaments are able to synchronize, because they need to be synchronized. And it has to be faster than the fluid. In this model, we have coupling between the fluid and the fact that they are in a free swimming cell. But that was not enough. So we went back to the biology. Look at these structures. And they are physical structures that couple individual filaments on the sliding. So they might be coupled at the sliding level already. We put this in the model, and this is exactly what we found. That the computational time decays for them to synchronize, but much faster, in orders of magnitude faster. So it's almost like these cells are trying to bring as much as is possible with them, that they are less prone to changing the environment, so that they are robust to change direction, to make decisions. That is vital. In particular, this cell is more clever than sperm cells, because sperm cells, they only have to deliver genetic payloads, but here you need to find nutrients, you need to reproduce, you have the whole life cycle. So the complexity, and everything relies on how you beat. If you go to the wrong place, you're dead, so you need to be able to find, oh, there's too much light here, there's not, temperature, so many other things. So it looks like that internal coupling is really the way that people are doing this. And if I may have time, I just want to finish with one thing, if that's okay, because then it connects with a question that I had at the beginning about, I've been talking about a lot about chemistry being important and self-organization coming from bottom up, right? And this idea, yes. But is that essential? Then what we did was we moved to muscle systems now. So myosin system. So there's something that is called the sarcomere that has a particular structure in which molecular motors, again, will bind to nearby filaments and pull the sarcomere, and this is how you have a contraction. This is how you can have a contraction. And it's known to have, if you put between these muscles with an elastic spring, they self-organize and they self-oscillate. Then the chemical cycle here is very complicated. It's also not well known. So we said, can we just say we don't have chemistry? Can we just say I have a mechanical motor that rotates, and every time this rotation interacts with a teeth, then you have an interaction, a binding happening. And when it doesn't, you don't have any binding event. And this is essentially a Kuramoto model. But a physical Kuramoto model, because we can also build this in the lab and make this like as long as this is in the same geometry as you find the muscle. Remember, my initial idea is that molecular motor, their actual molecular motor kinetics is not important. I believe more that the structure is what is important in having this level of self-organization. And then what we find, this is the system. It's self-organized. We have traveling waves. This is a myofibril, and these are the oscillations you get in a real muscle between two pipettes. And you get these deflections. And this is what also we get with a motor system that has nothing to do with that. Nothing to do with that. The only connection with that is that we put in these oscillators, these Kuramoto oscillators, in the same geometry as you would otherwise be in muscles. And because we get similar behavior, the conclusion from that is that this kind of behavior is not unique to chemical motors. At least we can conclude that. It's not unique. And if it's not unique, that means that there is a much bigger space of solutions that we could explore. But what's the good thing about this, especially for roboticians, is that it's very difficult for you to manufacture molecular motors. But anyone can find an electric motor. So what we're making is an artificial muscle type of unit that can replicate some of the organic properties that you see in real muscle. And then the applications on that, they're really amazing because now we're working in more miniature versions of that. From a mathematical perspective, this is also very beautiful if you are interested in the dynamic of networks. Because what you have here is a time variable network in which each node will only be coupled for a short period of time through the backbone. And also among long distances, because this backbone here is a stiff structure. So if I have three motors engaging, even if it's here and the other one is really far, they will be seeing each other. So you have a network, that's a time variable network, in which you have this blinking connectivity. And when there is self-emergence, you have the bands of connectivity that basically travels, through your network. So you can explore from that perspective as well. It depends on what's your background. I think I'll stop here. But there are more things. If you're interested, there are things in the fluid mechanics side as well, especially on the motion, on the fluid mechanics of, how can I say, of these organisms in 3D. So these are experiments where are done in Mexico by our Mexican collaborators, where they are able to reproduce the motion, like get the motion of sperm cells that beat in 3D and rotate in 3D. So really complicated. Then with the waveform, then I can solve the fluid mechanics and find what kind of flow fields this cell is actually producing. So I'm using here now fluid mechanics as a measure to measure something from the data from the experiments. And then this is also showing a number of other things, including that the fluid mechanics in low viscosity is not important. The forces are very small, even when you have like a boundary, things like that. But I don't know, I have no idea what time is it, but I can.
So, for the
Oh, yeah. Oh, let's stop. Yeah. Yeah. So there is.
We have a lot of questions, so maybe because we have much together, we can do the questions at once.
The questions from.
Oh,
Thank you.
there's questions
I have
online.
questions online. Yeah.
We have questions online. No. Okay. Thanks so much. And I think lunch is already waiting for us.
Thank you for people online as well.